there's also a piece of this that's about who is it that is now engaged in biology and life sciences. It's an incredibly diverse set or representative set of the world's population. It is people from all walks and backgrounds, from all parts of the globe. This is a picture taken at, maybe you can read, the opening of BIO. That's the Biotechnology Industry Organization's big annual meeting. This is biotech, extravaganza. And it's thousands and thousands of people. You can just look and tell they're from all over. They have all kinds of different interests and reasons for being there. What might be telling is something that you might be able to read up on this banner. This is the same meeting. Our only agenda is the success of the industry. So for these folks, it's about business and making money, and of course, at the same time, making products that are useful to people. So there's a big economic driver here in what's going on in the life sciences. And I don't want to suggest that there aren't some very important public health goods coming out of this. There are in spades. And you could pull up almost any issue of any biomedical journal these days and find a very useful discovery that's being described. It's a direct reflection of these abilities I've been telling you about with respect to generating diversity, designing things, packaging things. This is the novel production of probably the world's most important anti-malarial drug, a drug that was in limited supply because until this, it could only be produced by extracting it from the bark of the yew tree, which grows primarily in China, limited numbers of trees left. And now suddenly, Jake Kiesling up at Berkeley engineers a yeast with all of the complicated pathways for making a totally non-natural product in this cell. And you can crank out stuff now with orders of magnitude lower cost. So just to summarize, what's going on and why it's a little bit different in the life sciences than the physical sciences, because you've been thinking about where sciences have moved and where the threats arise, focusing on nuclear threats as one example. Well, here, there's a very different set of requirements for who it is that can do this stuff. The requirements are far fewer. It's much easier to gain access to the life sciences than in the physical sciences. The infrastructure you need is much less. And the kinds of things that might qualify you as a life scientist are far broader. There's the economic issue, and then basically the global nature of the enterprise. This is it's quite striking. So just a few examples of things that I've picked out, not because they're entirely representative of anything and everything going on in biology, but I picked them out because I'm thinking that you may realize, as I have when I think about this science, that this kind of science I'm going to show you is particularly important for the risks that it poses as well as the benefits. So in some ways not representative of all, for sure. First is the current ability to take the sequence of a viral genome and then make the virus de novo in the laboratory with chemicals. This is not all that new. This will seem old to some of you. This is 1994. This was the first reconstruction of the rabies virus. It was not the first reconstruction of any virus, but one of the important disease-causing viruses. 1994. This is basically, you give me the code, in a computer file, and I will then order a company or a service center to make me the DNA that encodes the RNA that I can then use to put into a cell and make rabies. And this is not true for almost all of the RNA viruses. Remember, there are two kinds of viruses: RNA viruses, DNA viruses. Influenza RNA, rabies RNA, smallpox DNA. So herpes virus, DNA. It's very easy for RNA viruses. It's becoming more and more easy for DNA viruses. This is an interesting story that came out just a few years ago. Ralph Barrick and Mark Dennison are at University of North Carolina and Vanderbilt, respectively. They study SARS. Do you remember the Sars Coronavirus? Really important new respiratory virus that came out of nature, out of some animal reservoirs, caused a worldwide pandemic that fortunately was contained and then suppressed several years ago. The question is, where did the virus come from? These folks looked at all of the current SARS coronaviruses and similar viruses, looked at their sequences, created a family tree, an evolutionary tree, and then walked back on the tree and said, if this tree is correct, there should have been, some years ago, an ancestor that gave rise to today's coronavirus, and we think it would have had this sequence, although no one has it today, right? So they actually ordered the sequence up from a sequence synthesis company and made that virus. So they basically resurrected an alleged ancestral virus today in the laboratory, and it worked. It grows, it has the properties one would expect. So this is a sort of an art and a science that now is becoming increasingly routine as a way of trying to understand bona fide threats to health from nature, possible would-be threats from nature, and anything else. It's also got important commercial applications. This is a related technology where you take a bunch of sequences and you cause them to recombine together at a high rate. You create a huge amount of diversity. And now you can create, for example, a new virus that instead of infecting the liver, which this virus on the top normally does, it now infects the heart. Or a retrovirus like HIV that normally infects, let's say, a primate, and now it infects a mouse. So you can really start to shift the behavior of these things around, partly as a way of understanding how they work, partly as therapy. That's a gene vector up on top, but think about the broader consequences. So another story from recent, or two stories actually, from the last couple of years. Two different scientific groups, two scientists, engaged in work on the surface of it for very useful purposes, they say. They stumble upon a result that is potentially problematic, and they deal with it in two different ways. And we can talk about that afterwards. Here's one story. It has to do with an influenza virus, a bird influenza virus, avian influenza, which in some birds doesn't cause any disease, in other birds causes disease and death. And it's spreading around the world, has been for the last 15 years, actually going back to the middle 90s. H5N1 is a bird flu virus, which, again, is well adapted to birds. Now, it has jumped into humans on a number of occasions by accident. People who are handling these infected birds in the marketplace, mostly in Southeast Asia, have infected themselves with this virus. And when that happens, the people get very sick and they often die. In fact, 60% of the people die. The good news is, not for these people, but for their family and others around them, the good news is these viruses, although they're great at spreading between birds, they are really bad at spreading between people. So if it gets into a person, it doesn't spread to another adjacent person very easily, if at all. And influenza viruses are known to be highly changeable. They mutate quickly. And so a lot of people have said influenza virus ought to have learned how to jump from people to people, person to person by now. It's been 20 years. Why not? So the U.S. National Institutes of Health said, we think this is an important question. We think it's important because we need to know whether it could happen. We need to know, if it happened, what the virus might look like and behave like, so that we could recognize it early or maybe treat it. So that was the rationale. And they said to several groups, we'd like you to try to make this virus. And so two groups took them up on this, and both succeeded. They took different approaches. a group led by Ron Fouchier in Rotterdam started with that actual highly virulent bird flu virus, which, as I say, causes 60% death in humans, but can't go from human to human. They started with that, and they engineered it in several different ways to try to give it that one thing it's missing, which is human-to-human transmissibility, or mammalian transmissibility. This other group took a different approach where they said, let's take just a piece of this bird flu virus, put it into a more benign viral genome, mutate the piece, but in this more benign background, and see if we can give the piece some properties that we predict would have given its natural parent virus the same new properties. So as I say, they both succeeded. And both works were published last year after a great deal of controversy and discussion. The crux of the matter was not that one shouldn't announce to the world that it can be done, that this virus does have the ability to acquire this new capability, but rather, should one publish the exact genetic sequence of this new engineered virus? Because influenza is an RNA virus, and it falls into this little dictum here. If I have that sequence, and I am reasonably savvy in influenza biology, I can make it. I don't have to go through what Ron Fouchier did and evolve it myself. I can just take the sequence, order it, make it in the laboratory. And some of you may have read about this whole story. It was actually a lead editorial in the New York Times 2 years ago. It was called, a Doomsday virus, an engineered doomsday. And so the kind of discussion that went on was an attempt to undertake a risk-benefit analysis, which is a tough thing to do. Well, some people tried, and they talk about benefits. Some of them are here, and they talk about risks. Some of them are here. And how one balances this out depends upon who you are, how much of this you're familiar with, whether you're able to critique these technical features adequately, and then almost how risk-averse you might be, depending upon where you see this leading and when. But one of the questions I think was not so well discussed is, what about the timing of benefits and risk? Suppose that we accept these risks, which I believe are real, which is, if this thing were to get out, could we contain it quickly? And my answer is no. Most public health people, I think, would agree. The answer is no. And if it even had 1/10 that case fatality rate, let's say it's 3%, not 30, that's already worse than the 1918 influenza virus. The pandemic was probably the worst pandemic of the last 100 years. Because that virus was only 2% case fatality, and it killed many tens of millions of people. So I think we accept there are some big risks here, but the point about timing is you've bought them right now. Because as soon as you release that sequence, in several months' time, somebody can produce that virus. Well, the benefits may be real as well. We understand what it looks like. Perhaps we can detect early versions of it sooner. Perhaps we have a better angle on developing a vaccine and drug and so forth. But how long would that take? You've got the risk now. These benefits, in my estimation, are probably years off. And it's actually more than just making drug and making vaccine. It's got to be delivered. Remember 2009, this pandemic influenza we had? That was actually a very close relative of a known influenza virus for which we have vaccine. So as soon as that 2009 virus appeared, people went to work like crazy to make new vaccine for this virus, new virus. And it was done pretty efficiently, but it didn't happen in time to stop that virus in 2009 from going around the globe. So I don't think we even have the public health infrastructure to deliver something that might be available today or in several months' time in order to stop the spread of a pandemic flu virus. So then you've got another issue. If everyone who might work and resynthesize this virus were somebody that we understood well, had a good working relationship with and understood their working conditions, that might be one thing. But there's a lot going on in biology, and I don't mean to sound at all critical. I think a lot of this is really cool. Actually, they think it's cool too. Because the purpose of Science Hack Days, just as one example, is to build cool stuff. And these are happening in, in sort of big, empty spaces, basically. Meeting places, halls, gathering places, where people get together, share their thoughts, some rudimentary equipment, which is now pretty powerful, and are starting to tinker with DNA. Want to make an E. coli that will detect a pesticide that you might care about in your food so that you could have the organism sitting on a plant and glow when a pesticide hits it or something. It's interesting, somewhat kind of difficult ideas, but nonetheless, people are doing this sort of thing. And remember that the more people you have doing something, the greater is the likelihood of an accident. Now, these are not people who intended for SARS to get out, but it did. It's happened about 3 or 4 times.